Howdy, folks. Thanks for checking in to MrUlrichsLandofBiology.com. I am Mr. Ulrich. In this little notes cast, we're going to be talking about cell membranes and the life process of transport. Remember, of course, transport is a life process, something that all living things do, and it involves moving material here and there, wherever it needs to go. In this case, we're talking about moving material in and out of cells. The diagram back in the background here, I have a little cartoon strip, I guess. Here are little particles of something or other. It doesn't matter what they are. What's important is that they can get through that cell membrane. Right here in the beginning, there's far more over here than there are over here. And those particles then, because they're bumping into one another, right? They're bumping into one another like a break in pool. That means that these little things are going to be spreading out, trying to get further apart from one another as they bump into one another. And if the cell membrane is permeable to them, and it looks like they certainly are, if the cell membrane is permeable, those particles are going to spread out just like that cell membrane really wasn't there. Until over here, they're in equal concentrations on either side. Here's a good old amoeba. As any living thing going through cellular respiration, it's going to be producing lots of carbon dioxide. So the carbon dioxide concentration within the cell is going to be continually increasing. As a good aerobe, it's continually using oxygen. So the concentration of oxygen is continually going down within that cell. Hopefully for that cell, it finds itself in an oxygen-rich environment, and likewise in a carbon dioxide-poor environment, so that the oxygen will tend to diffuse back into the cell, and the carbon dioxide will diffuse out of the cell. Now, what are we talking about here? We're talking about getting material in and out of cells, that life process of transport. And there's really two categories of this. I'm focusing on one right now, and that's what's called passive transport. In the case of this amoeba, it's not doing any work to get the carbon dioxide out and the oxygen in. It's just kind of flowing out of it by the action, by the movement of the molecules of carbon dioxide and oxygen spreading out. So that's expending no energy. We call that passive transport. There's also active transport, and that's when the cell actually uses energy to move material around. We'll get to that. Passive transport, moving materials across that cell membrane without using any energy to do this. The primary mechanism is going to be by diffusion. The only mechanism, really, is going to be by diffusion. Diffusion, as I've been describing, is when material moves from a region of higher concentration to a region of low concentration. When things spread out from a place where they're kind of packed together to a place where they're not so packed together. Now, anytime you have a difference in concentration like this, we call this the concentration gradient. In your lungs, in the air inside of the lungs, hopefully you have a very low carbon dioxide concentration in the air that you're breathing, especially compared to the carbon dioxide concentration in the blood that surrounds the lungs. Because you want that carbon dioxide to move from the high concentration of the blood down the concentration gradient, we say. Carbon dioxide doesn't always move down into the airspace within the lungs. So that happens by diffusion. And is an example of passive transport. Water, kind of important to living things, is so important that the diffusion of water, when it moves from that region of high concentration to a region of low concentration, gets its own term, osmosis. So when we're talking about osmosis, we start out with some water, some watery solution in an environment. We're going to say it's in a beaker. And in this beaker, we're going to chuck an amoeba, a really, really huge amoeba. In this case, I don't know why. So our two regions are the beaker, which is the environment, and the cytoplasm. In this beaker, we're going to put a 4% salt solution. The solution has two parts. It has the stuff that makes up the bulk of the solution, which is the solvent, in this case water, and the stuff that makes up the least part of it, called the solute, in this case salt. So I have a 4% salt solution. The cytoplasm, we're going to say, is a 2% salt solution. Now, it's not just salt. There's this stuff, other stuff that's in there. It doesn't matter what that other stuff is, but for our purposes right now, we're going to say it doesn't matter. All right, the important thing that we want to focus on here is that there is a 2% solute solution in the cytoplasm, and there's a 4% solute solution in the environment. The terms that we use to refer to these relative concentrations of salt and solute are hypertonic, for, in this case, the beaker, because it has more dissolved stuff. Hyper is for more, so it has more dissolved stuff in it. And hypotonic, which is for less dissolved stuff in it, we'll say. Now, the weird thing, of course, is we're talking about osmosis here. We're talking about the diffusion of water. And actually, the salt, for all intents and purposes, does not diffuse across the cell membrane. So we're not really focusing on the salt. What do we care about? We care about the water in this situation. So the terms kind of refer to the solvent concentration indirectly. So in this case, the beaker then has a 96% water solution, and the cytoplasm is a 98% water solution. So there's your concentration gradient. There's a higher concentration of water inside of the cell than there is outside of the cell, and water tends to move from the region of high concentration to the region of low concentration. So the water is going to leave the cell, and that cell is going to get smaller and smaller and smaller and smaller and shrink. Now I can take that same cell with a 2% salt solution, 98% water solution, and put that into a beaker now with only a 0.5% of a solute in it. That makes it 99.5% solvent, pretty close to pure water. Now, that same cytoplasm that was hypotonic before is now hypertonic, and the beaker is hypotonic. So these terms are relative. I can't give you a beaker of water and say, is this hypertonic or hypotonic? Why? Because it's compared to what? So in this case, now, since the beaker has the higher concentration of water, and the cytoplasm has the lower concentration of water, water is going to tend to move from the environment into the cytoplasm. So cells in hypotonic solutions in freshwater situations tend to take on water and swell up. Now, if you're an animal cell, you're going to swell up and pop, right? Because the cell membrane, remember, those phospholipids are not bonded to one another. So there's, they can't take a whole lot of pressure, osmotic pressure. But plant cells and other types of organisms that have a cell wall, a rigid cell wall, they can withstand some of that osmotic pressure. And that pressure builds up within them. It makes them turgid, is the term. They're swollen with water. And it makes them nice and plump. Now, the last situation that we haven't talked about, we're going to take this 2% salt solution. We're going to put it in of a cytoplasm and put it in a beaker of 2% salt solution. In this case, they're isotonic to one another. There isn't one with more or less dissolved stuff. So now the water is in equal concentrations on either side of the cell membrane. That does not mean that the particles, in this case water, stop moving. They do move, they just move in equal amounts from one side to the other. All of these situations, hypertonic, hypotonic, are going to proceed until, they're going to try to proceed until both of the solutions are isotonic to one another. And at that point, they're going to continue to move from one side to the other, but it's going to be in those equal concentrations. In this case, in an isotonic solution, the cell is not going to change size. The cell's just going to kind of chill. So here's a weird question. If you have the opportunity to drown and be resuscitated, would you rather have that happen in the North Sea, which is cold and salty, or in the Waukil River, which, though right now it's a little chilly, is typically warmer than the North Sea, and it is certainly is freshwater, has less salt. Osmotically speaking, when animal cells are exposed to fresh water, like there is in the Waukil River, that's a hypotonic solution that's going to cause the cells to swell up. In the case of the animal cells, they're going to pop and lyse and break apart. But those same animal cells in the North Sea are going to lose water. They're going to desiccate. They're going to dry out. for all intents and purposes, that doesn't evaporate, but it through osmosis, the water diffuses out of the cells are going to shrink. Now, in the case of losing water, if I take those same cells and expose them to a biologically isotonic environment, those cells are going to reinflate. They're going to take water back on, and some of the damage is going to be reversible when it comes to losing water to the environment, where once the cell lyses, it's pretty much done. Passive transport is moving material without using energy. Then active transport is going to be moving material with using energy. One example of this is when the cell has to move material up or against the concentration gradient, that is from a region of low concentration to a region of high concentration. Diffusion is going to want to bring it the opposite direction, so the cell's going to have to pump it against the concentration gradient. One classic example of this is the sodium-potassium pump that is found in the cell membranes of your neurons. They're what makes up and maintains an action potential for nerve impulse travel, which we'll talk about later on when we talk about the nervous system. But I have a better diagram here next. This takes place through protein channels, transmembrane proteins that go all the way through the cell membrane. Remember, many of these make up those tubes through which substances can diffuse in some cases. In this case, it's kind of like a tube, but it's closed on one end. And here, the sodium transport tube is open to the inside and closed to the outside. And the sodium particles that are bouncing around inside of the cell in lower concentration but still bouncing around can get stuck to the inside of this transport cup. When ATP comes along, and that's the energy molecule that we'll talk about here in another couple classes, when ATP comes along and bonds one of its P's, lends one of its P groups there, to the transport cup, the transport cup snaps closed and opens up on the other side. And once it opens up on the other side, the sodium molecules also don't bond very well to the transport cup, and they get kicked out into the higher concentration environment. And then the opposite happens with the potassium. The potassium moves from the lower concentration outside of the cell to the higher concentration inside of the cell exactly the same way, by deforming that cup to the opposite direction. Another type of active transport involves freshwater organisms which find themselves in hypotonic solutions. So there's a much higher water concentration outside of their cells than inside of the cells. Paramecium is one such type organism that is constantly taking on water because of its hypotonic environment. These paramecium have adapted a type of vacuole called contractile vacuoles. These specialized vacuoles here, you can almost see them, they have these little tubes that radiate away from them, and these tubes pump the water right through the pellicle to the outside environment of the cell. So as the cell takes on the water from the outside environment, these vacuoles fill up, and then a change takes place in the proteins that surround them in the cytoskeleton that squeezes that vacuole small again and squeezes the water back out. Because that pumping actually takes action, takes energy, we call it active transport. Yet another version of the cell using energy to move material in and out of the cell. In this case, in is called endocytosis. Endo for inward, cyto for cell, cis, process for now. So this is an inward cell process. One version of this is called phagocytosis. Phagy for eat. And this is when the cell surrounds a big chunky bit, a big solid particle. It bumps into the cell, into this particle, and sends the pseudopodium to surround it. And remember, it's the cell membrane, since it's like bubbles. Those phospholipids are not bonded to one another, so they can, when they pinch off, it can form a bubble. In this case, within the bigger bubble of the cell. So that material kind of gets locked inside of a vacuole. Now it can interact with lysosomes, and that large chunky bit can be broken down, and its subsequent parts be absorbed through the cell membrane, actively transported, or likewise diffusion. Pinocytosis is another version. Pinot for wine, I believe, or at least drinking. In this version, instead of bumping into a large chunky particle, it's really just surrounding bits of the extracellular fluid and any of the dissolved particles that are in that extracellular fluid. Receptor-mediated endocytosis really is referring to how the receptor molecules can kind of play a role in determining whether those pseudopodia, whether invagination is going to take place. In this case, if the particles that the cell encounters fit the shape of the outside of their cells, fits with the receptor molecules, then the change in those receptor molecules translates into changes in the cytoskeleton, which cause a dent to form. We call this invagination. And that dent forms and kind of pulls in that material, and it pinches off again, bubble within a bubble, and now that material is surrounded within a vacuole. Exocytosis would be the opposite of this process. So material that's within, waste material that's within a vacuole can then be moved towards the cell membrane, where, like bubbles, can kind of become one and release their inner contents into the outside environment. We'll stop there for now. Thanks for checking in to MrUlrichsLandofBiology.com. If you didn't get there through my website, make sure you check that out so that you can get any of the accompanying notes sheets and all of the other good stuff that goes along with MrUlrichsLandofBiology.com. Of course, you can always drop me any questions or comments or concerns or cannolis to my email there. Other than that, we'll see you in class.